Cancer Council WA acknowledges the traditional owners of country throughout Western Australia. This recording was captured on Whadjuk Noongar country. We recognise the unique connection that Aboriginal people have to land, waters, sky and community, and we recognise elders past, present and emerging. Today I'm talking to Dr Scott Cohen, who is the Senior Research Officer at the Children's Medical Research Institute in the Cell Biology team. Our conversation aligns with Genes for Genes Day. Thank you very much for joining me today, Scott.
Thank you, Tim. Thanks for the invitation.
Now, Scott, I just want to set a bit of a standard for all of our listeners today. So very simply, can you tell me what cancer is?
Yes, in the most simple and general terms, cancer is a disease of uncontrolled cell growth. Now, to understand that more, it's useful to first look at, what is normal? What's normal cell physiology? And, if you look at us, sometimes we're symmetrical in some fashion. All of our organs are very defined proportions. And a fundamental and essential property of our cells is that they will divide a finite number of times. And of course, this is programmed into our genes. And another property of cells, of non-disease cells, is that they are responsive to and even dependent upon the neighboring cells in the tissue. And in biology, we sometimes call that the cell's social context. Okay? Each cell has its niche, which dictates sort of how it behaves. So with those properties in mind, if we think about cancer, at its root cause, cancer is a disease of genetic mutations. That's the underlying cause. And so it can be a very rare cell that has acquired a profile of genetic mutations that allows it to bypass these built-in limits of cell proliferation. And in the laboratory, we actually refer to these cells as immortal. Okay, cancer cells really do divide without limit. And referring to the cell's, social context, the other thing cancer cells do is they have disengaged from the social context, and they are no longer responsive to the neighboring cells and tissue. Okay, so a cancer really is essentially it's independent outgrowth of normal cells, and depending upon where that is in the body, it becomes dangerous and can kill you.
Your work in particular, Scott, has been around those finite divisions that should exist in an otherwise healthy cell. So tell me about telomeres and your work in that.
Yeah, well, started back in the early 90s, cancer researchers realized that understanding, cellular immortalization and the growth properties of cells meant understanding this field of research we call telomeres and telomerase. So telomeres are the specialized ends of our chromosomes. Okay, so remember our DNA chromosomes, think of it as a long piece of string. Each has two ends. So the ends of our chromosomes are kind of special, and those are what we call the telomeres. Now, what normally happens in a non-disease state is that telomeres shorten a little bit each time a cell divides. And eventually, the telomere shortening will proceed to a point that it instructs the cell to stop dividing. We refer to telomere shortening as nature's timepiece. Okay, it's what places those limits of cell proliferation. And it's a normal part of aging, both at the cellular level and at the physiological level. Now, where telomerase comes in, again, the cancer cell, it's acquired this profile of genetic mutations, and one thing it can do, it can turn on telomerase. So telomerase is, in science, we call it an enzyme. It's essentially a molecular machine in the cell. And what telomerase does, it replenishes the telomere length. Okay, telomerase lengthens the telomeres, and because there's now no telomere shortening, there are no limit of cell proliferation. Okay, so by maintaining constant telomere length, telomerase confers cellular immortality. Another property of telomerase with respect to a potential cancer target is that roughly 90% of all human cancers have reactivated telomerase. And so telomerase is found in virtually cancers of all different tissue types. And so from a molecular target perspective, telomerase has what we call broad scope, in that a therapy targeting telomerase could potentially be appropriate for almost all cancers. And that's a very efficient way of developing a therapy. I liken it to, you go to the chemist and get an antibiotic, a broad spectrum antibiotic you may have heard of, where, a single antibiotic would be appropriate for all sorts of different bacteria and infections. Okay, again, it's an efficient, effective way for therapy development. And so that's one of the promising potentials of telomerase. Another favorable property of telomerase is that for the most part, our normal non-disease cells, telomerase is undetectable. So if we can develop a therapy that targets telomerase, that should be more specific for the cancer cells and not affect the healthy non-disease cells. And in practical terms, specificity at the molecular and cellular level translates to a safer therapy with fewer side effects.
So, Scott, I just want to go back to something you said earlier around cancers existing outside of the social construct. And I know you don't have a medical background, but from your work, how do current treatments work on that cellular level?
Yeah, so, the mainstay of cancer treatment is still chemotherapy, which has been around for decades and, is still evolving. And, well, all chemotherapy targets the property of cell division in some manner. More relevant to the research we do is we're interested in the chemotherapies, many of which damage DNA. Okay, and it just so happens that cancer cells are more susceptible to the damage of chemotherapy than normal cells. Now, but I think as we all know, chemotherapy still lacks that specificity, and patients endure pretty significant side effects. And that's because the chemotherapy cannot wholly discriminate between the cancer cell and the non-disease cell. And if I think about cancer research in general, these days really the name of the game is specificity. Regardless of the many different treatments cancer researchers are developing around the world, the main aim these days is specificity for the cancer cell. That's going to give you a safer therapy.
So your work has been around more targeted intervention with fewer side effects.
And that's one virtue of telomerase. It's present in almost all cancer cells, but the vast majority of our non-disease cells, telomerase is undetectable. So the hypothesis has been for a couple decades now that a therapy targeting telomerase should display fewer side effects than classical chemotherapy. So specificity for the cancer cell will translate into fewer side effects throughout the body and a safer therapy.
Which would mean much better patient outcomes, much more
Exactly.
targeted therapy, less hospitalization, all
Quality
those
of
side
life,
effects.
and exactly.
So it all sounds incredibly promising, Scott. What is the process from the laboratory into clinical practice?
Good question. Yeah, it's a long process. In the laboratory, I just want to emphasize, any therapy that one might be taking now, there's at least 2, possibly even 3 decades of fundamental laboratory research behind it. I've been working on telomerase for 22 years now. So I've done my 2 decades of fundamental telomerase research, which has led to the identification of a small molecule that blocks telomerase action. We call that a telomerase inhibitor. And right now it's at the point of being a really great research tool. Okay, so in the laboratory, it works in a test tube. So biochemistry, working with telomerase in a test tube. And we've also now applied it to cancer cells that we can grow in laboratory culture. And from there, the next step is what we call preclinical animal models. So mice, usually, that have been, these are specialized experiments designed to assess the efficacy and to some extent the safety of a new therapy in an animal model. And I must emphasize, it's a model, but we call this generally preclinical. And efficacy in these models is essential before the next step would be first in human phase 1 trials. So clinical trials are generally divided into 3 phases. So a phase 1 trial, really the only question is, how safe is the therapy? Can we put this thing in a human body without any, deleterious toxicity? They're not necessarily looking at efficacy, although sometimes that can be measured too. It's really not until phase 2 where the safety has been established, and then with greater numbers of patients, they really focus on looking at the efficacy of the new therapy. And a phase 2 trial, it's often very focused, a single kind of cancer, okay, to obtain a very definitive conclusion. And beyond phase 2 is phase 3, where now a much greater number of patients and a much more rigorous test of efficacy. And so the few challenges of getting into that, that first phase 1 human trial, first of all, you need very compelling preclinical data. Now, the other challenge isn't so much scientific, it's financial, where, you're looking at 10 million, possibly even $20 million. And it's at this point where usually you need to partner with a company. Now, some people sort of make their own company and raise sort of capital funding that way, or you can partner with a more established biopharmaceutical company. But the bottom line is, so first of all, that you need the financial resources, and also as a independent academic institution that we are, much like a university, they're not in a position to accept potential liability of a clinical trial. Remember it, clinical trials and experiment. And so there's financial and also very legal accountability challenges that must be established as well. So there's a lot of science behind it, but then there's these other factors that must be overcome as well.
Scott, yourself and Professor Tracy Bryant were both the recipients of some Cancer Council New South Wales funding. How has that assisted in your research so far?
Yes, so it's been very crucial. It's provided us stability and continuity in our research. No secret, research expensive, it takes money. What's very important also in scientific research is the stability and continuity. I mentioned the time scale of research. It's years, even decades. And now the grant cycle is often, every couple of years, sort of never ending. But the reality is, a major research project, will cover multiple grant cycles, and so to have that stability and continuity to continue the research is very important. Now, Cancer Council, generally, as a not-for-profit institution, is crucial because the competitive grant schemes that most scientists pursue through the federal government, in particular, there's the National Health and Medical Research Council. That's the federally funded research grant scheme. Unfortunately, those have become more and more competitive over the years. Rather than government funding increasing over the years, it's gone down and is continuing to go down. Now, your listeners there might be surprised. I think most people I speak to think, we write our research grants, we submit them, we get the money, and off we go. Your listeners might be surprised that from several years now, only 1 out of 10 biomedical research grant proposals have been funded by the federal government. Okay, if I flip that on its head, 9 out of 10 research applications for funding have been rejected. And it has almost nothing to do with scientific quality. Most of these are sound science. It's essentially, it's poor funding at the federal level. And this is where these nonprofit institutions like Cancer Council come in and fill the gap and are really crucial for scientists like me to give us that continuity and stability for another few more years, until we can try the application process again. So I might take this opportunity, if your listeners, they might be surprised at how challenging funding is. I would say to the listeners, write to your federal parliamentarian and demand an answer to the question, why are 9 out of 10 biomedical research proposals being rejected? That's a very fair question. And that is not my opinion. That's a fact. And it's been that way for several years, unfortunately.
Scott, you spoke really wonderfully about the importance of receiving grant funding. Now, I want to ask about your organisation, Children's Medical Research Institute's flagship day, Genes for Genes Day. What's the importance of it, and how does it help supporting research?
Yes, Genes for Genes is it was still is our flagship kind of fundraising campaign. It started in 1993. At that point, Tracy Bryan, my colleague and partner in crime here, she was at CMRI as a PhD student studying cancer cell biology and telomeres. And it really became, I think, the key identifier for CMRI out in the community. It was really it still is a grassroots fundraising effort. It's a means to identify CMRI out in the community. And the money that has been raised through these grassroots community, generosity has been absolutely crucial to now, decades of research. So it's been over 30 years now, raising a significant amount, which has subsidized the research programs, just like funding from Cancer Council. Ultimately, it comes from fundraising and community donations, and so we're very fortunate to have that generosity of the community to support our research. Again, when, for example, if we don't get a grant, we have this fundraising that can carry us over until the next cycle and give us that stability and continuity of our science. If we always want to think big as scientists. Having that security to do so is important.
Scott, do you want to highlight any particular outcomes that have come from these donor funds?
Let's see. Yeah, some of the experiments I did to initially identify this telomerase inhibitor. Goes back about 10 years now. Sure, a lot of these funds are directly from Genes for Genes fundraising. Pretty much all of our major research programs at the Institute have benefited from Genes for Genes. So our program into, very broadly, we call it neuroscience and neurobiology, which has implications for epilepsy. We have a very strong gene therapy program, which has the potential to provide a curative therapy for inherited genetic diseases, for which currently there may be no treatments. So yeah, Genes for Genes has positively affected pretty much every research program in the building for 33 years now, I believe.
It's wonderful to hear. And we will certainly be promoting Genes for Genes heavily throughout. And we'll make sure we put some references down in our show notes so that people can head on over to the website and contribute as well if they're able to. In my team, Palliative and Supportive Care, we are always talking about quality of life and improving quality of life. And someone who doesn't have a research background, how much thought is put into the outcome and the impact it's going to have on people when developing these new technologies or exploring these new ideas?
Yes, at the scientific level, we know that the deleterious side effects of not just chemotherapy, but many treatments, at the molecular and cellular level, it comes from poor specificity in that the therapy is not discriminating adequately between the cancer cells and the normal cells. And so in cancer research, there are many areas of cancer research beyond telomeres and telomerase, but I think the underlying theme for all of those researchers is to develop a better and more targeted specific therapy, specific for the cancer cells, which will reduce the side effects and the burden that cancer patients have to endure during treatment. I'm very local. I walk to work, and my walk to and from work takes me through the Children's Hospital, which is next door to our building. I walk through the playground and the gardens behind it, and around the corner from the oncology ward, and walking through a children's hospital twice a day is the ultimate reality check. And it's that kind of constant reminder of we have more to do, and just how much things can improve.
It's great to hear, Scott. Now, Scott, you have been working on this project for 22 years now. How do you remain connected? You just said that you get to walk through the pediatric hospital and get to see it firsthand, but how do you remain dedicated for a program for this amount of time?
Yeah, that is a long time, and it's the longest I've worked on any molecule, telomerase. A few things. So, as I just mentioned, walking to and from the kids' hospital, sometimes you get a very harsh reminder of there's still an unmet need. Another way, I'm not ashamed to admit this, at the day-to-day level, I'm a science geek, and scientifically, telomerase is just stunning. And there's so many discoveries that are still waiting to be uncovered. And for me, that also, walking into lab, I'm giddy with excitement each day. That's what science geeks are like. And then another kind of way we stay connected. For a long time, we have a close relationship with a patient advocate. This gentleman came to CMRI about a decade ago. He had just lost his wife to cancer, and he wanted to be proactive, not just in donating funds, which he has, but also just learning about what is cancer. So he's not trained as a scientist, but he just enthusiastically jumped in and tried to learn about the science, not just with us, but also at another cancer lab at another university, I believe UNSW, University of New South Wales, across town. He regularly attends our research lab meetings, and to his credit, he's actually absorbed a lot of the principles of cancer cell biology and telomere biology that we focus on. But nonetheless, he will often ask that pointed question that only a patient advocate who's been through cancer firsthand, really, that they'll think of. And so that is also a good reminder that just keeps us grounded, and again, reminding us that we still have a long way to go.
Scott, what do you wish people knew more about cancer research?
I wish people would understand the time scale of scientific research. And although it seems slow and it's often one step forward, two steps back, nonetheless, what scientific research gives us is hope. And so research it's slow, it's frustrating, but I feel confident in saying that over time treatments will improve. Whether it will be my telomerase inhibitor, I don't know. It's too early to be sure. Okay, this is still research, but we know through decades of scientific research that things will improve, and they are continually improving. I hope people realize that research is equivalent to hope. And I guess my other wish is well, this may not be directly relevant to your listeners, although they could potentially have an impact. I wish research was embraced more in this country in terms of funding and its importance in so many aspects of society, and in particular cancer, but many aspects of our quality of life. So yeah, that's those are my wishes.
Scott, I've certainly learned a lot about telomeres and a lot about research and how it comes into fruition and what that progression looks like. And I really appreciate your time for joining us today.
Okay, thank you, Tim. And yeah, it's been fun. Hope your listeners have enjoyed that.
Thank you very much, Scott, for joining us today.
Anything referenced in today's episode will be captured down in the show notes. You can follow us on social media platforms, LinkedIn and Facebook, or you can head over to our website, pace.com, for any education or training needs that you have. Catch you in the next episode. Bye.